learning objectives include why we stain microorganisms and what is acid-fast staining. As I mentioned earlier, stain or staining procedure enhances colors, and human eyes appreciate colors, and that is the reason we can see organisms more clearly. Now, this acid-fast staining is a kind of differential staining because there are only certain microorganisms that have this capacity that once they're stained, they resist decolorization or destaining with acid alcohol. Paul Ehrlich observed this phenomenon that when he stained Mycobacterium, he observed that when he used acid alcohol for destaining, the microorganisms retained the stain. They did not decolorize. So that led to this procedure, which now we call acid-fast staining. In this procedure, the background of this procedure is that Mycobacterium microorganism has in its cell wall a waxy material called mycolic acid. This makes the cell wall impermeable to most stains. And the trick is, again, we need to heat the stain in order to get through the cell wall. Needle method is the most commonly used method, in which you make a smear and then you flood the smear with carbulfoxine and then heat the slide along with the stain for 5 to 10 minutes. And after that, you wash with water and decolorize using acid alcohol. Then next procedure is counterstain, because those microorganisms that have mycolic acid, they would retain the original stain, carbulfoxine. But that do not have mycolic acid, they would be destained. So in order to see them, we use a different color which we call a counterstain. In this case, methylene blue. For 1 minute, you stain with that. And then after washing the slide and air drying, we see under the microscope. And you will see if the organism is acid fast, it will look pink. Many of my students get confused with this color, because if you remember in gram staining, pink color basically tells us gram negative. These acid fast Mycobacterium basically is gram positive. So remember that this is a gram positive. Here it is looking pink because of a different procedure of staining, and that is acid fast staining, not because of the gram staining. So if you look at the gram with the gram staining, it will look blue. So keep that in mind, please. So in summary, acid fast bacteria have mycolic acid that can be stained by heating the organisms with the stain. And those who have mycolic acid, they resist destaining with acid alcohol.